We're going to be talking about the structure of DNA and how that structure affects the function of DNA. And the two things that we're really going to focus on are being able to identify the pieces. So you were working with physical chunks of stuff, like things that look like this or this the other day. I want you to know what these are called. And then those hook together to form other pieces that have names, and I want you to know what those are called. And then more importantly, so those names, those are things that in the past, like when I was in biology, not just your age, but in college, the expectation was we would memorize all of this stuff. So that's why I have it all memorized. But the reality is, memorizing things isn't nearly as important for most people now as it used to be. There was an advantage to memorizing things before we all had like Google in our pockets. So now what I want, and the reason that I'm going to have you take notes, is I want you to have this information accessible in a place where you can find it easily. So when it comes to identifying the basic structures of DNA, this is more of a information access. I want you to have it written down so that later on you can get that information when you need it, and you will. This piece here is a little bit different. The structure of DNA, the way that DNA works, the way that it hooks together, ends up determining two incredibly key parts of the way that it works. The function of DNA is related to its structure. And that's a really common refrain in biology, structure and function, structure and function. We talked about this before when we were talking about enzymes. Enzymes work because they physically fit their substrate. The lactase enzyme physically fits the lactose sugar and allows it to break apart a little bit more easily. It's physical. And it's chemical too, because the fit is held together by chemical bonds between the enzyme and its substrate. So DNA works the same way. DNA is tiny. We tend to think of DNA as being like in charge, right? Like something about these things like tell your body something. And we haven't really talked about it too much, but DNA is like does something really important in your body. So DNA is in charge, but all of the DNA in your body isn't even a cell. It's just inside your cells. DNA is just a chemical. It's just a chemical substance. The fact that it does what it does is entirely a product of the fact that it tends to hook together in a certain way. And that's it. There's no more to it than the fact that it happens to piece together in a way that makes it do two really critical things. And we're going to look at both of those. Let's start at the beginning. So we've talked a little bit about this because when we were talking about biochemical compounds, this came up. DNA is a kind of nucleic acid. So I do want you to know that when we think about our four different biochemical macromolecules, there were carbohydrates, proteins, lipids, and nucleic acids. DNA is one type of that fourth kind. There's other kinds as well. DNA is kind of the premier. That's the one that we think of the most. When we think of nucleic acids, we think of DNA, but there's other kinds as well. You do not need to know this name, deoxyribonucleic acid. You don't even need to write it down. That's the D and the N and the A of DNA is all. And DNA is a chain of nucleotides. Again, this is something we talked about when we talked about biochemical macromolecules. We talked about polymers and monomers. And what we said was nucleotides are the monomers that go together to make nucleic acids. And so a nucleotide is a piece of something. If you take one nucleotide and hook it to another one, and another one, and another one, and another one, you get a nucleic acid. And if it's a certain kind of nucleic acid, then what you end up with is DNA. DNA is a bunch of repeating chunks, and we're going to look at those chunks today, and we're going to look at the pieces of those chunks. Those chunks store genetic information. But what's that? We all know, like from like casual conversation, like genes are like, well, that's why my hair is this color and your hair is this color and why Grigar is bald. And that's why like some people are taller than others, or some people have some fingers that are longer than others. And that's all genes, but what is a gene? A gene, really simply put, is instructions for proteins. Genes are sequences, pieces of DNA that encode instructions for making a protein. And it gets really, really difficult not to insert an overseer into this. And we talked about this before, the idea that it's almost impossible for us to think about an instruction manual without somebody to read it. But there is nobody to read it. There's nobody reading the DNA instruction manual. It all happens just on its own. And we'll talk about that on a variety of different levels. We'll talk about it on the most straightforward level today, but we'll continue talking about it in more and more detail over the next couple of weeks. So what that means is you have these nucleotides hooked together, and if they go together in the right order, then somehow in there is the information needed to make a variety of different kinds of proteins, including things like the lactase enzyme. The lactase enzyme, the instructions for it are in your DNA. And proteins are used for a couple of different things. We talked about this when we talked about biochemical macromolecules. They are functional in that they can sometimes just speed up chemical reactions, control the rate of reaction, or they can be structural as well. Like your hair is made out of protein, and that is a structural thing. It's not reacting with stuff. The last key piece of information to think about with DNA is that any time a cell reproduces, the resulting cells need to have the same DNA as the original cell. If we're looking at hey Lucius, Luke comes up here. If we're looking at a single-celled organism like a bacterium, then in order for that bacteria to reproduce, it needs to split in two. And the DNA that was in the original bacteria has to be in both of the new ones. For multicellular organisms like us, just to grow, our cells need to divide. If I cut my hand accidentally, then new cells will form to heal that cut. All those new cells need DNA. It needs to be the same DNA that was in the original cells, which means the DNA needs to be copied. So we've got two things that we have to think about with DNA. One is, how does it encode information? And the other is, how does it get copied? Again, there is no overseer. If I were to give you a set of instructions like this and say, oh hey, we need another copy of this, you'd be like, well, I'll take it to a photocopier. If I said, well, the power's out, and photocopiers aren't going to work, you'd say, well, I'm going to take a picture with my phone. And I might say, well, no, we need hard copy. We need this on paper. You could still make a copy of this with a pen. You could just like write it out. And you'd write it out, you might make a few mistakes, might not look this good, but basically you could get the same information. But the thing about that is that when you make a copy, a person makes a copy of something, it goes through your brain. Your brain is like looking at stuff and telling your hand what to do. There is a control center that makes that copy. Imagine taking this piece of paper and throwing it out into the woods, and 2 days later coming back and finding two pieces of paper that were identical. It just like made a copy of itself. There wasn't anybody to do it. There wasn't an overseer taking charge of it. That's how DNA copies itself. There's nobody in charge. It just happens. And that has to be the way that it works, because this is all happening on a subcellular level. So we're going to talk about these two things today. These are going to be our big pieces today. And then we'll dive into both of these more on Thursday. So we can think about the pieces of DNA by thinking about the idea of a monomer versus a polymer. So a monomer is a single piece of something. A polymer is what happens when you take several of those pieces and hook them together. In DNA, the single piece is called a nucleotide. This right here is what a nucleotide looks like. It's all one word, but I couldn't fit it on there. So a nucleotide has some different pieces to it. there's 3 parts, and you've seen all 3 of these parts. So I'm going to show you in these drawings, and I'm going to show you using the model. One of those parts is a sugar. It's a special kind of sugar. It's called deoxyribose. It has 5 carbon atoms in it, but it's a carbohydrate. It's a sugar. It's got carbon, hydrogen, and oxygen in certain proportions, just like lactose, just like glucose, just like sucrose and fructose. All those sugars that we've talked about already, this is just a different kind, a different sort of sugar. This piece right here in your models is the sugar. The piece that had 5 gray dots, spheres, and 2 red ones. The red ones are oxygens, the gray ones are carbons. So if you look at this picture right here, this is our sugar. And the name of the sugar, and you don't have to know this, but the name of the sugar is deoxyribose. Just like lactose, fructose, glucose, sucrose, if it ends in O-S-E, it's a sugar. So this is what it looks like. And another piece of a nucleotide is called the phosphate group. In your model, this was the phosphate group. The yellow piece is a phosphorus atom. It's an atom of phosphorus. The red are oxygens. And this right here is our phosphate group in this picture. And the last piece is what's called a nitrogenous base. This is the nitrogenous base in this drawing. And in your model kits, the nitrogenous bases, there were several different kinds. They were these pieces that had blue and gray and white on them, and sometimes a red. So there were 4 different kinds that you came across, and they have letters A, C, G, and T, and we'll talk about those letters in just a minute. When you take these and you hook them together chemically, you get what's called a nucleotide. In your model, those chemical bonds are the strongest bonds in DNA. So if I take my deoxyribose sugar and I look at the sort of the socket that's got like a curved top to it, I can fit one of my nitrogenous bases into there, and then that's pretty sturdy. That's not going anywhere. And if I look at my phosphorus group, there's sort of a socket for that too, another piece that fits right in. When this is all hooked together, this is really, really sturdy. This doesn't come apart easily. In fact, some of you probably noticed when you were taking your models apart, you had to like kind of figure out where the connection was. You can't even really see it when you're just looking at it. You have to like kind of pull a little bit to discover where it comes apart. This is one nucleotide right here. If I hold it up against the screen, the sugar is down here, the phosphate group is up here, and the nitrogenous base is here. DNA is a polymer of nucleotides, and that means that they're all hooked together. They get hooked together in 2 ways. The most important way is that this piece right here, this chunk that consists of the sugar and the phosphate group, can get hooked to another sugar and phosphate group on either side. That makes what we call the backbone. If we look at this image right here, we can see that backbone right here. Here's a phosphate group and a sugar. Here's another phosphate group and a sugar. That could potentially continue, phosphate group, sugar, phosphate group, sugar. The nitrogenous base sticks out this way. When you were working with your models, many of you discovered that you can do that by just repeatedly hooking these sugar and phosphate groups together in a repeating pattern. So it's very straightforward how these all get hooked together. They hook together the same way, and you can see the pattern. Like it visually, they hook up the same way. And one of the things that you probably noticed was that when you hook a phosphate group to a sugar phosphate group that's already hooked together, you hook it with a magnetic connection. it's a little bit weaker. It's a little bit easier to pull that apart. And that is true of an actual DNA molecule as well. The connection between this phosphate group and this sugar is strong. The connection between this sugar and this phosphate group is a little weaker. And that's true everywhere we have that connection. There's one strong and one weaker. The strong one tells us what the nucleotide is. This is one nucleotide, but the nucleotides can be connected to each other. Those magnetic connections with the model are a little bit weaker. And so we could continue doing this infinitely. There's no limit to the number of connections we can make. And this right here is what we call the backbone. This is the backbone of a DNA molecule. And then the information in DNA is actually stored in these nitrogenous bases. There's 4 different kinds. We use letters as abbreviations, and we'll look at what those are. But if you think about the way that this is laid out, and you think about how the nitrogenous base is attached, you realize you can put any nitrogenous base in here in any order. So this one is an A, and I can put it at the top. I can grab a random one. I don't know what this is. This is a G, and I can put it right below it. And this, I don't know what this is either. This is another A. I can put that right below there. And I can add any nitrogenous base, and I can do it in any order. Here's another A. So I just happen to have an A, and then a G, and then 2 more A's. And I could keep going. I could assemble any length with any sequence. And that's the information. The sequence of these nitrogenous bases is the information. And the fact that we have 4 to choose from, and we'll look at this in just a second, and that we can put them in any order means we can encode as much information as we want to in this. We could make like sequences of DNA to stand for every word, every number, every letter, every concept in the entire universe. We could make a unique sequence of DNA to represent every atom in the universe. It would be possible to do that. Now, it would be ridiculous, but it would be possible. This is a 4-letter infinite word code that can store an infinite amount of information. And it's this easy to put together. The way it goes together is critical to the information storage. If we think about a single strand of DNA like this, we can think about reading these in sequence. And you don't need a person to do it. You don't need an intelligence to do it. You just need something that can recognize the chemical signals of these 4 different nitrogenous bases. Yeah?
So with bigger creatures and smaller creatures, would the size or the amount of the genes and DNA be different?
Not necessarily either, actually. The amount of DNA roughly corresponds to the number of different kinds of proteins, but larger creatures don't necessarily have more kinds of proteins. They've just made more proteins. So you can think about it like Legos. Like if I were to look at building something out of Legos, I could make something huge out of all the same Legos. It doesn't take a lot of DNA to make this particular Lego. If I make something small, but it's made out of a bunch of different kinds of Legos like this, I might need more DNA.
So like a blue whale would even...
I don't know. So that a blue whale doesn't necessarily have more DNA than a human. I don't know though. It might have more
Would the DNA
You
be-
can't just look at the physical size of the organism though.
Would its cells be bigger?
No, not necessarily. No, they might be, but probably not. Most animal cells, like on average, are about the same size. But a blue whale, what it does have is more cells. They might not be bigger, but there's way more of them.
Yeah, because it's a lot bigger.
Because it's a lot bigger, yeah. The 4 kinds of information that we can store in DNA, we give it letters. You only need to know the letters. Most of you probably have this memorized already from like your past biology classes, but just in case you don't, one of them we refer to as A, one of them we call C, one of them we call G, and one of them we call T. Those letters are the names of the chemicals themselves. each nitrogenous base is a similar but not identical chemical. The A stands for adenine. The C stands for cytosine. The G stands for guanine, and the T stands for thymine. You have to know the letters, but you do not have to remember what the actual name of the bases are. You should be able to look them up. Like if I, at any time, were to say, hey, what does the C stand for? I would expect that you could grab a book or grab your notes or grab your phone and find out that C stands for cytosine, but it's not critical that you have that memorized. Yeah?
So did they just shorten it so it's easier to say?
Exactly, and easier to write down. Yeah. And then if you look at these, you can see 2 of them are kind of smaller and 2 of them are kind of bigger. And the larger ones we call purines, the smaller ones we call pyrimidines. You don't need to know that either for this class. You should, again, be able to look it up. Ideally, what I would hope is that down the road, if somebody mentions, oh, well, some of the nitrogenous bases are purines and some are pyrimidines, there would be a little blip in your brain that goes, oh yeah, I remember we talked about that in bio. And you don't have to remember what it is, but you can look it up. Interestingly, I can never remember. I never remember which are the purines and which are the pyrimidines. I don't remember which ones are bigger and which ones are smaller, and I don't remember which letter is which, because it doesn't really matter for what I do. But for the next couple of weeks, I will remember that A and G are the larger ones, and those are purines. C and T are the smaller ones, those are pyrimidines. I'll remember that for the next couple of weeks, and then it'll be gone. So it's okay to have a short-term access to some of these things. The structure of DNA, when you put it all together, it's not just a chain that consists of the phosphate group and the sugar as a backbone and nucleotides coming off on the side like this. Because it turns out that, that for a whole variety of chemical and physical reasons, it always comes in 2 strands. And those 2 strands get woven together and twisted together. You'll see what that looks like in just a second. That is what we call the double helix. So the double helix of DNA consists of 1 strand that looks like this, and another strand that isn't identical, but it matches up to this. It attaches to it on the other side. You saw that when you were building your molecules the other day. This is a single strand of DNA. Here's one sugar phosphate group, here's another sugar phosphate group, here's another sugar phosphate group, here's the nitrogenous base on each side. This is the backbone of 1 strand of DNA. If I take this and combine it with another strand and then kind of twist it up, I get that double helix. The sugar phosphate backbone is both sides. So if I look at the twisted shape here, one side that's twisted up is made of sugar phosphate. The other side is also made of sugar phosphate. The backbone, every DNA molecule essentially has 2 backbones, 1 on each side. This is only half. And the nitrogenous bases form what we think of often as rungs of a ladder. And the rungs of the ladder pair up in a predictable way. And we'll see why in just a second. Those rungs of the ladder hook together so that everywhere there's an A on one side, there will be a T on the other side. A, by the way, is a purine. It's one of the larger ones. T is a pyrimidine. It's one of the smaller ones. And everywhere there's a G, it will get paired with a C. G is a purine, one of the larger ones. And C is a pyrimidine, one of the smaller ones. And it kind of makes sense if you think about it, because if you have 2 different sizes, you have to pair a larger one with a smaller one for everything to fit evenly. You can't put 2 larger ones together and then 2 smaller ones together, because it's not gonna line up right. And these connections between the base pairs are called complementary. Complementary with an E, not with an I. Complementary with an I is what I would say if, for example, I was feeling pretty good about Caroline, and I were like, Caroline, you're doing great work today, I'm feeling good about you. If that were true, I would say that. Of course, it isn't true, so I'm not gonna say it. You're not gonna hear me say it. That's a compliment. A compliment is when two things go well together. So when I make a peanut butter and mayonnaise sandwich, no, I'm just kidding.
Like complimentary colors.
Yeah, like complementary colors, yeah. I can't even eat peanut butter. I wouldn't put it with mayonnaise anyway. But complementary flavors, complementary colors, that's with an E. And in this case, what it means is two things that go together, that fit together. And here's what's kind of crazy about this. When you think about how these two strands of DNA have to match up, it seems like what you would have to do is kind of look, right? Like it seems to me like I would have to be like, okay, well, this one is an A, so I better find a T to line up with that. But that's not what happens. Instead, what happens is we start with nucleotides. And those nucleotides potentially are just floating around loose inside of a cell. In the case of DNA in larger, more complex organisms like us, this would be floating around in the nucleus of the cell. And when this nucleotide comes close enough to this strand of DNA, it's going to match, but it's only going to match where it fits. And it's only going to fit where there's a A. So you don't have to think about it. It just isn't gonna fit unless it's the right base. And that's true for every single base pair that we could look at. It's not something where there's some sort of creature or being or thoughtful anything that's looking at it going, I know where this goes. It's a matter of, we have a nucleotide and it's floating through the cytoplasm and it can hook, but it's not gonna hook in the wrong spot, it's only gonna hook in the right spot. This is a G, it's gonna match with a T. And the same thing is gonna happen again. Uh-oh, I'm missing a piece here. Some of these have gotten slightly mauled. Okay, I need. Here's a T. So this floats along and it encounters another A. And now that these things are next to each other, these can hook up as well. And so what ends up happening is you get this much more sturdy shape. And I'm going to add my last nucleotide because I want you to see how this all works when it gets coiled up. So I'm going to add my last A nucleotide, or sorry, T nucleotide. And now I have, this is the double helix. In this configuration, it just looks like a ladder. Here's sugar phosphate backbone, here's sugar phosphate backbone, here are my complementary base pairs down the middle. But if I twist this, and the models are designed to do this, you can see it takes up a little bit less space. And that's what happens typically in cells, is that the DNA twists up like this to form not just a ladder shape, but a twisted ladder shape, and that's the double helix part. When these base pairs match up, when you use your model, you'll see it's magnets that hook them together, and that's because the connections between the base pairs are hydrogen bonds. Just like the hydrogen bonds that form when water molecules weakly bind to each other. It's not a very strong connection. It can be relatively easily broken apart. So that's one of the reasons why DNA, we think of a DNA strand, a single strand of DNA, as being a thing. It doesn't have to come in 2 strands, it just usually does. It's a lot sturdier like this, but it can come apart. And these connections, A with T and G with C, purine with pyrimidine, pyrimidine with purine, were discovered and named by, I think he was Russian scientist named Chargaff. You don't need to know that, but that's one of those things where down the road, somebody might ask, have you ever heard of Chargaff's rule? Now you have. So you can forget it, but don't forget that you once knew it. And so this is what it looks like when it all goes together. Here are some sugar phosphate back connections with their associated nitrogenous base. These are hooked together. The nitrogenous bases hook across the double helix to another nitrogenous base with hydrogen bonds. The whole thing gets twisted up. When you look at a diagram like this, what I want you to see is this. And this, there's a reason that we don't typically draw it out like this. It's harder to see. This makes it a little bit easier, but even this is a little bit difficult to see. So oftentimes we do stuff like this where we just have like a shape and we say, well, that's our adenine, and we have another shape and we say that's our thymine, and they sort of fit together to indicate that the physical and chemical connection between these two things is why they fit together in DNA. And so here's just another view again. Here's our ladder view with backbone, nitrogenous bases. They could potentially split apart. The weakest link is between the nitrogenous base pairs. So it typically would split into 2 strands like this. And this is what it looks like when it's all twisted up. This is what it looks like if we use little spheres to symbolize the atoms, and that is almost exactly what you'll see when you are working with your models today. Is there's slightly different colors, obviously, but pretty darn close, and then it gets twisted up like this. And we have the same, effectively the same drawing, only it's a lot easier to work with it when it's in 3 dimensions. And then the last thing that I want to just point out is where the DNA is. So in prokaryotic cells, bacteria and archaea, it's located in an area in the cell, but it's not in a membrane. But in eukaryotic cells like animals and plants, it's in the nucleus. So the DNA is in the nucleus of eukaryotic cells, and that's primarily what we're going to deal with. So as we think about DNA, one of the things that tends to happen is people tend to just kind of think about it as being big because we're working with these models and stuff, but the reality is it's not just small, it's smaller than any one individual cell. It's smaller than the smallest bacterial cell because it has to fit inside it. All right, so what I want you all to focus on today is exploring how it is that the structure of DNA lends itself to A, storing information, and B, being copyable without somebody being in charge of it. And so there's some instructions in the activity. The first thing you're going to need to do is rebuild a model. And then the second thing you'll need to do is go to the instructions and start going through, and it's if you follow them step by step, it'll help you see how this all works. You're going to take your double helix, and it should have at least 5 base pairs in it. You're going to take it apart. You're going to give half of it to a different group, and you're going to keep half of it. And what you will see is that if the other group just kind of pretends that this is floating around in the cytoplasm and has new nucleotides come and attach to it, what you'll see is that they'll end up creating this strand. And that is the key to DNA replication. We'll look at that in a little bit more detail on Thursday, the mechanics of it and where you start and where you end. But for today, what I want you to do is just like get it embedded in your brain that this is a byproduct of the structure of DNA. It's not something that anybody needs to think about. Now is a great time to take a break. So it's 2:20, come back here at 2:25, and let's get started with the models.